Okay, so today we are going to start the microsatellite lab, where you will be interpreting microsatellite data. So in every organism's DNA, there are repeating sequences of bases. So when we talk about bases, we're talking about those genetic nitrogenous bases: adenine, guanine, cytosine, thymine, right? The A and the T, the C and the G. We've talked about those before. And these occur in different lengths, and these can be used to identify individuals. These regions are called microsatellites, and they occur in regions of DNA that usually do not code for proteins. So proteins are enzymes. We just recently did a whole lab on, and we're actually not done with it, on lactase, right? Lactase is an enzyme that breaks down lactose, which is milk sugar. And lactase is an enzyme, but it's also a protein. And proteins are a specific sequence of amino acids. So in this lab, we're going to be looking at regions of DNA that don't usually code for proteins, okay? And we are going to analyze them. They are cut out of the DNA, isolated, and then copied using polymerase chain reaction. So polymerase chain reaction is a process where if you only have a small amount of DNA, you can make copies of it. It's kind of like making photocopies of it. This is very useful in crime scenes when there's very little forensic evidence. They can take a small piece of forensic DNA and make copies of it. It also tags these small pieces with a fluorescent marker, and then they are fed through tiny glass capillary tubes in a machine that actually reads the tags. And these microsatellites show up in sequencing software as little peaks that indicate the length of the repeated segment. So the higher the peak, the bigger the segment of DNA. The smaller the peak, the smaller segment of DNA. And since chromosomes come in pairs of 2, it's possible for one individual to have 2 different microsatellites, meaning 2 different alleles, or they can have 2 of the same. So if they're exactly the same, this is called homozygous. The prefix homo means same. And if they are different, then that is heterozygous, because the prefix hetero means different. So you are going to use the microsatellite sample on the next page to fill out your data table with the analysis of the individual mountain lions in the sample. And then you're going to answer the questions. So the first mountain lion is RF009. And so we're going to go down here. Oh no, sorry, I have it on a different paper for you guys. Okay, so we're going to go over here. So here is RF009. So look at here. At marker 109, you have 2 peaks. And at marker number 137, how do I know it's 137? Because this is 135. This is 135 right here. And then this is 140. So each one of these lines is a marker. So this is 137 there in the middle. And then over here, this one's at 109. So let's go back to our paper. Allele from parent 1, 109. Allele from parent 2 is at 137. And because they're different, that's heterozygous. Okay? I'm going to show you another one, RF012. So let's go back to that. We're going to go down here, RF012. Okay, see how there's only one marker, and that's at 117? That means these two are exactly the same, so it's 117, 117, homozygous. I am going, and then you can see again, they gave you another one, RF 493, 117, 127, heterozygous. So if the two numbers are different, they're heterozygous. If the two numbers are the same, they're homozygous. I'm going to do one more with you, and then you're going to do the rest on your own. So let's go to RF 010. So look at, we have 117, 137. So I'm going to come back here, and I'm going to write, whoops, what just happened? Here we go. I'm going to write 117, and we're going to move that. Oh, I hit it too quick. 137. I'm going to move that up here, and then I'm going to write heterozygous. Okay, so I want you to pause the video, and I want you to fill in the rest of them. I'm going to give you 5 minutes to do that. Ms. Gupta is going to give you 5 minutes. Okay, you can work with a partner. I want you to fill in the rest of this chart. Okay, here is what you should have gotten. Ignore the two little numbers on the upper left. I tried to line this up, so please take a moment and check your work. You can pause the video and then continue. So now to some questions. Why might scientists collect microsatellite data? You know that this is chromosomes, right? So you're looking at the relationships between the chromosomes. So, and then you're looking at them between individuals. So they're primarily used as markers for genetic profiling. So let me see if I can get something to write with here. Not that. I need the text box. Okay, there we go. Used as markers for genetic profiling. So what we mean by that is we can call this kinship studies. So how related? I already told you that they're used in forensics, but we don't need to mark that here, but I'm just saying that's another use. And what do you notice about the animals in the samples that you looked at? Were most of them homozygous or were most of them heterozygous? So hopefully you said most of the, whoops, there we go. Got to get another thing here. Most of the animals were heterozygous, meaning they had different alleles. All right. Moving on. So now we are going to look over the mountain lion genotype data and see what we notice. Okay, so the genotype data should be on the flip side of the microsatellite data. So you can see you have individual populations. You've got 5 members, right, from California, 5 members from Mexico, 5 members from South Dakota, Florida, and Texas. And you can see their markers. So I want you to take a moment and see if you notice anything. Okay, so you really have to know what to focus on. So what I'm going to show you is Florida and Texas are probably the most closely related. They have the smallest distance in their allele frequencies, meaning like the 106 is generally either 244 or 246, where up here you've got 250s and 254s thrown in. Here, another one, same thing. You've got 240s, 246s. There's a couple of 254s down here. Your A312, one allele is like 270s to 280s. Here, 270s to 280s. Here, 207, B207 is in the 300s. And then again, 316 is in the 250s to 270s. So these two are the most alike when you're comparing the chromosomes. The two that are the most different would be Florida and Mexico. So if you look at this line, you've got 250s and 248s. This is 244, 246. Over here for 312, you have 260s and low 270s. Here you have 270s and 280s. Up here you've got in 207, this is all 320s. There are no 320s down here in Florida. And in the last one, this is like 262 basically and 264. And this goes basically from, it does go from 262, but it's 266, 274. So these are the most diverse. So that's kind of hard. Oops, going the wrong way. That's kind of hard to look at. So we're going to go back here. So what do we notice? Let's see. We notice that Florida and Texas are most similar. Shortest distance in frequencies. And Florida and Mexico are the most different, and they have the largest distance in frequencies. Let me fix my spacing here. Okay. So you need to find something that somebody else wrote, okay? Pair, write something one that somebody else wrote here. And so you got to get two ideas from other people, like what they found. Okay, I'm not giving that for you. I'm not in the classroom. You got to talk to your peers. All right, now it says, analyze the mountain lion genotype. Using mountain lion genotype data, list the alleles that occur in each population in the data table. Only list each number once. These are called unique alleles. We will do California as a class and then split up the rest. Okay? Okay. So what we're going to do next is I'm going to do California with you. So I'm going to highlight. So what do we have? We have 250, we have 254, we have 248. Those are all different. Is there anything over here? We have 254, so 252. Okay, so those are the four. You're going to write those four down for 106. Okay? So we've got 250, 252, 248, and 254. All right, now for gene 312, we have 272, 274, 276, and 280. So we got 272, 274, 276, 280. Those are the ones that are different. Then for gene 207, we've got 310, 304, 324, 318, and 330. So again, we've got 304, 310, 324, 318, and 330. And in the last two, for gene 316, we have 266 and 268. That's it. Those are the only ones that are different. So you're writing those down on your document. I'm going to do these with you so that it doesn't get messed up. Now we're going to go to Mexico. Mexico, we've got 248 and 250. All those are repeats. We got 254. So we've got 248, 250, and 254. Everything else is repeated. For gene 312, we have 266, 272, and 274. You're writing these down on your data chart. And then for gene 207, we have 322, we have 326, and 328. And then for gene 316, we've got 260, 262, 264, and that's all that's different. All right, going to South Dakota, we have 246, 250, 254, 258, and 252. Again, 254, 246, 250, 258, and 252. Going to gene 312, we have 266, 270, 272, 274, and that's it. All right, in gene 207, we have 302, 316, 324, 304, 306, 308, and we already did 324. That's a lot of diversity there. 302, 316, 324, 304, 306, and 308. And in gene 316, we've got 258, 262, and 272. 262, 258, and 272. All right, going to Florida. 244, 246. That's it. Going to gene 312, 270, 280, 276. That's it. All right. Going to gene 207, 308, 314, 324. That's it. 314, 308, 324. And going to gene 316, we've got 262, 274, 266. That's it. 262, 274, 266. And Texas, we've got 246, 244, 254. That's it for Texas in the first gene, gene 106, 246, 244, and 254. In gene 312, we've got 272, 276, 278, 270, and 280. So that's 270, 278, 276, 272, and 280. In gene 207, we have 308, 314, and 324. And then in the last one, we have 258, 266, 262, 272, and 274. So 258, 266, 262, and 274. So write them down in your chart. I am not going to write them in the chart because it's going to take too long for me to type, but you should have them written in the chart, because now we're going to come back to this, right? Actually, I'm going to see if I can read ahead and stay on this page. Okay. So once the chart is all filled in, right, these are what's called unique alleles, because we've only listed the number once. Now they want us to examine our list of alleles and circle the numbers that only appear once in a population. And these are called private alleles. So now we're going to come back and we're going to find well, okay, so 254, 252, 248. Okay, so all of the ones that I've written down in this one, let me just double checking. These are only appearing once. Hold on, let me just double check the instructions. Okay, I had to reread the directions. So now we're looking at individual columns, right? So we're looking at this 106 gene. And are there any numbers that only appear once? Like 244 is here, but it's here also. 246 is in several places. 250 is in several places. 254 is in several places. 250 is in several places. So there's nothing in this first one that is only in one population. Then when I go here, 252, that appears here. 254, that appears here and here. 258 doesn't appear anywhere else, so I'm going to circle that. But you're not circling it on the data sheet, you're circling it on your worksheet. Now I'm going to go to the next one, 276. 276 is down here. 272 is down there. 274. 274. 274 only appears once. Okay. What about 266? Nope, that's more than once. 272, that's more than once. What about 270? 270 is more than once. 280. Do I see any other 280s? I don't see another 280 in this column. Okay, 276, I think we already said. Yep. 270, we said. 278. Do I have another 278? We have another 276. We have another 272. I don't see any other 278. Okay. Now we're going to 312. So 280, 280, 274, 274. Okay, 310. Do I have any other 310s? I do not have another 310. 324, I have another one. 304. 304, I have another one. 302. I do not have another 302. And I don't believe that I have another 316. Okay. 324, I have another one of. 304, I have another one of. 306. Do I have another 306? I do not have another 306. Okay. 314. 314. I have another 314. 308. Do I have another 308? I do have another 308. And a 324. I have another 324. Okay, what about 326? I don't know how I missed that. I don't see another. I don't see another 326. I'm just double checking. And what about 322? I don't see another 322. Where did that go? I think I got them now. All right. Gene 207, 308, 30, nope, sorry. 318 is only appearing once. 330 is only appearing once. 328 is only appearing once. 308 only once. And 324. All right, 316. I got 262. That's there. 266. 258. Another 262. 274. 274 only appears once. We did 266, 258, 262. What about 264? Any other 264s? I don't see another 264. And 262 is a repeat. 266, 260. Do I have any 260s? No. 268 is... I don't have any 268s either. And 266 is a repeat. All right, last column, just 272. Whew. So we have all our data. We just circled the numbers that appeared in only one population. These are called private alleles. Now we're going to compare the mountain lion genotype to the Connecticut cat. So here is the Connecticut cat. This is the Connecticut cat gene information, and we're going to compare that to the mountain lion. So the row of data above is from the mountain lion that was hit by the car in Connecticut. Compare this to the alleles, the unique alleles. So the unique alleles were the ones that I highlighted in green. Those are the ones we first wrote down, not the ones we circled. And what do we notice? So I want you to compare these numbers, okay? Take a minute and do that and see if you can notice anything that's similar. So you should be figuring out where this Connecticut cat gene sequence most matches, okay? Which population? California, Mexico, South Dakota, Florida, and Texas. Well, you should see that it matches the most with South Dakota. Because if you look at the South Dakota data, right, I want you to look at it right now, and I'll read you the Connecticut cat. 246, that appears in the first column of South Dakota. 258, that appears in the second column of South Dakota. 266 appears in the third column. 274 in the fourth column. 302 in the fifth column. 324 in the sixth column. 262 in the seventh column, and 272 in the eighth column. So this so it matches most with the South Dakota data. All right, so that's what you should be writing there. Okay, next we have a See, Think, Wonder. You're going to use the graphic organizer below to record your ideas from this investigation. So things you see in the data collected, think, ideas that this data makes me think about, and wonder, questions and wonderings that you have about the data. Okay, so some things that we've noticed in the data that we've already talked about is that the Connecticut cat is most closely related to South Dakota. And another thing is that we noticed, we said it earlier, Texas and Florida are more closely related. Mexico and Florida are not closely related. These are things that we already stated. I'm trying to get that to fit in the box, so to speak. Okay, those are some things that we see in the data that we collected. So what does this make us think about? So it makes me think, is the Connecticut cat from South Dakota. And also did the mountain lions in Mexico or Florida do something similar? Meaning, did they disperse to find a new mate? So that's something there. And questions and wonderings do you have about the data that you need to fill in on your own? Okay. I will give you a few minutes to do that. We'll give you 5 minutes to get this down and to write any additional questions or things that you're wondering about in this last box. You should write at least one thing, and then we'll look at the analysis questions. Okay, so where do we think the Connecticut cat came from? Well, we already said this, right? We feel that the Connecticut cat came from South Dakota. So the Connecticut cat came from South Dakota based on genetic analysis of DNA markers. Also, there were actual sightings of the cat along the traveled route. Okay. So that's what we've concluded. What level of evidence, individual cats, population ranges, genetic information, populations that you've studied so far was most helpful in figuring out where the Connecticut cat came from? Well, obviously, you can write what you want, but I think that the unique unique markers were the most helpful because they linked the Connecticut cat to the South Dakota cat. How do we think the mountain lion got to Connecticut? We talked about this at the beginning. I don't know if anyone remembers. He got there by dispersal, so traveling to find a new mate. Now, we know that they don't usually travel that far. He traveled over 1,000 miles. It actually took him a year to get there. Number 4, based on what you know so far, why do you think that some populations had many private alleles while others did not? So this isn't probably something that you would know yet, but it has to do with small group size, isolation. So small group size and isolation, or it can be recent rapid expansion, which all prevent the exchange of genetic material. Okay, and there's one more question. When studying populations, it is important to collect samples that accurately represent the larger population. The data below shows the approximate population size of each location. So you've got the 5 different populations we studied and their population size. 20 mountain lions were collected from each of the locations above and studied in order to gain data about the population of the lions living there. Based on the population size data, which sample from which region would you expect to be the least accurate to represent the population as a whole, and why? So which one of these would be least accurate? Hoping that somebody in the room is saying Florida and not just sitting there waiting for me to do all the work since I've already passed the test. All right, Florida. Why Florida? It's because it has the smallest population size, which would not be representative of the entire population. Okay. Get that written down. Okay, so to finish up, I am going to read to you the full article that appeared in the New York Times regarding this incident, which it was a true incident. So from the beginning, the tale seemed to skirt the edges of the possible. A mountain lion reported to be stalking the wilds of Greenwich, Connecticut, seen near fancy private schools and busy roadways. But the sightings were confirmed in early June by paw prints, photographs, and animal droppings, and then, more dramatically, by the lean body of a 140-pound male creature killed by a sport utility vehicle on the Wilbercross Parkway in Milford on June 11th in 2011. The aftermath was even more surprising. On Tuesday, in what state officials termed amazing news, they said the Connecticut cougar had made its way east from the Black Hills of South Dakota, and genetic testing matched samples of an animal confirmed as having been in Wisconsin and in Minnesota. That means that the animal traveled more than 1,500 miles to Connecticut, more than twice as far as the longest dispersal pattern ever recorded for a mountain lion. The news stunned researchers trying to make sense of this first confirmed presence of species in Connecticut in more than a century. Many believe that the animal must have been released or escaped from captivity. Daniel C. Etsy, Commissioner of Connecticut Department of Energy and Environmental Protection, said the journey was a remarkable and positive reminder of the ability of wild animals to survive and adapt, but that there was no evidence that the mountain lions were returning to the state. This is the first evidence of a mountain lion making its way to Connecticut from western states, and there is still no evidence indicating that there is a native population of mountain lions in Connecticut, he said. But the finding may add at least a smidgen of mystery or paranoia to dozens of reports of similar creatures in Connecticut and the Northeast, most of them investigated and then dismissed as mistaken impressions. Before the animal was reported seen in early June in Greenwich, the last confirmed sighting of a mountain lion in Connecticut was in the late 1800s. Dennis Shane, communications director for the department, said that the development could lead people to wonder if there were other mountain lions, but that there was no reason to believe so. So at this time, you are going to turn in your paper and you are going to work on the Ed Puzzle karyotyping assignment if you did not finish that from yesterday.